Hello again, everyone, and thank you for coming. And again, thank you to the organizers, Dr. Zaid, for inviting me. It's been a wonderful visit so far. The people are so friendly and welcoming, and I'm enjoying my trip very much. So, before we get started, on behalf of the Poultry Science Association, I do want to invite everyone to attend the first Poultry Science Association Pacific Rim Scientific Conference, which will be held in Macau, China, in October of this year. And there's a QR code here. You can also talk to me about it afterwards. So we welcome you to join us in Macau. So I feel a little bit out of my depth here because usually I'm talking to nutritionists, but obviously in a room such as this, where there's many virologists and immunologists and microbiologists, I'm probably going to be telling you things that you already know. But I wanted to give this context because it's important to understand how nutrition interacts with the immune system. And so when we think of the immune system, we think of it as having two different arms. So first of all, we have the adaptive immune system, which involves targeted responses. And this is things like antibodies and directed cytotoxicity mediated by T cells. And so this is a very specific system. It also is, it has memory. So this is what we're trying to induce when we vaccinate. When the adaptive immune system is activated, it's a low energy cost. So nutritionally, there's not a lot of requirement to support activation of the adaptive immune response. Now, because it's very targeted, it tends to have very little collateral damage. So there's very little damage to tissues of the host cells and tissues after activation of the adaptive immune response. Now, obviously, there can be abnormalities where the adaptive response attacks the host's own tissues, but we're talking in normal circumstances. The challenge in poultry, though, is that the adaptive immune response typically takes some time in order to really become fully functional. And so this graph simply shows that when chicks are challenged at 0 days of age or 4 days of age, they don't really respond to certain antigens. And it's not until they get a little bit older, when they're challenged, say, at 8 days of age, that we start to see an adaptive response. And so this is very important because the chick then hatches with a relatively immature, poorly functional adaptive immune system. And so the chick needs to be protected from the potential pathogens that are present in the environment. And so we have the second arm of the immune system, and this is the innate immune system. The innate immune system includes barrier functions, simply keeping pathogens out, which comes at a cost. But also, there are some active portions of the innate immune response. So things like inflammation that involve phagocytosis and killing of potential pathogens. Now, we don't typically think of the innate immune system as having memory. There is some work showing it can be trained, but not typically what we would think as memory. And so every time a particular challenge is faced, there's a similar response. And so it's not as efficient as facing multiple instances of the same kind of challenge. The innate immune response, particularly inflammation, can have very strong local effects, but also very strong systemic effects. And that's where we really start to see that interaction with nutrients and nutrient requirements. So the innate immune response, particularly inflammation, tends to be energetically costly, and that's what I'm going to spend a lot of my time today talking about. There can also be collateral damage because the mechanisms of inflammation are not specific. It involves creating inhospitable conditions, generation of free radicals, generation of molecules that can not only destroy the pathogen, but can also damage the host tissues as well. And because the adaptive immune response is very slow to develop, or relatively slow to develop, the innate immune response is particularly important in the early stages of a bird's life. And if we're talking about broilers, for example, these birds might be going to market at 35 days or 42 days. So they're very young. And so the innate immune response is very, very important for a large part of the bird's life. There's also communication between the innate system and the adaptive system. So in order for there to be a good adaptive immune response, it is necessary first to have a good innate immune response. So the two systems work together. And so typically what we see is that when there's a challenge, there's an initial innate immune response involving inflammation. And gradually, as that innate immune response progresses, in many cases, the innate response then transitions into an adaptive response. And so when we look at how do we minimize the negative aspects of inflammation, one of the ways that we can do this is see a more efficient or more rapid transition from an inflammatory response, which is energetically costly, to an adaptive immune response, which is much less nutritionally expensive. So one of the things I tell my students when I'm teaching about the digestive tract is that the lumen of the digestive tract is actually outside of the animal. So food and other components of the feed can go through the digestive tract, but it's not really part of the animal's body until it crosses that intestinal barrier. So the physiology and the anatomy of the digestive tract involves a huge amount of surface area. So we have villi, and we have microvilli that increase the amount of absorptive surface area, making nutrient absorption much more efficient. But that also creates a lot of potential exposure for that tissue to the outside environment, including to bacteria, to commensal bacteria, to pathogenic bacteria, to viruses, to protozoans, and basically anything that's in the digestive tract. So the digestive tract has one of the largest exposed surface areas to the outside environment. And so when we think of the function of the digestive tract, maybe there's two conflicting goals. So obviously when we think about the digestive tract, we think about letting things in. So taking nutrients from that outside environment, the lumen of the digestive tract, across the intestinal barrier and into the body, into the bloodstream, or into the lymphatic system in the case of some birds. And there are active and passive processes to bring nutrients from the lumen of the gut into the tissues. But at the same time, there's an incredibly important component of intestinal function that involves keeping things out. So non-nutrients and microbes, including the commensal or normal bacteria and pathogens. And so there's a barrier function there, but there's also an active immune response. So there are antibodies present, there are phagocytes present, there's pathogen killing, there's immune sensing of what's going on. And when we lose that barrier function, or when microbes can overwhelm the ability of the digestive tract to limit translocation, then we start to run into some problems. This is some work done in Brazil. This is a meta-analysis published. And what these researchers did was they looked at a large number of research studies, 174 different papers. There were 183 experiments involving over 120,000 broilers. And so what they did was they looked at, in these experiments, the effect of removing antibiotic growth promoters. And so overall, they found that there was an increase in the feed conversion ratio, a decrease in productivity, and essentially it cost about 3 US cents per bird. And so they extrapolated this to the entire Brazilian industry over the course of 1 year. They calculated that it was about $183 million in lost revenue or additional costs because of the removal of growth-promoting antibiotics. And so it's clear that growth-promoting antibiotics work at what we ask them to do. But we're obviously in a world and an environment where there's government pressure, there's consumer pressure, and I think scientific pressure to change the way that we use growth-promoting antibiotics and move away from growth-promoting antibiotics. So the inflammation that occurs when particularly intestinal immune system, and if there is translocation of microbes, systemic inflammation comes at a cost. And so we see that in the previous slide with reduced efficiency and increased costs of controlling disease. Now, those costs are largely associated with the inflammatory response. So we can look at the inflammatory responses having effects at the local tissue level, so at the intestinal level, but there are also systemic changes that take place. And so the combination of these two responses can really have a huge impact on the growth of the birds, the efficiency of the birds, and ultimately on productivity and profitability of our poultry operations. This was some work done, and researchers looked at two environments. So a very clean university type research facility, and then in that same facility, they had an unsanitary, basically a dirty environment. In each of those environments, the birds were fed either a control diet with no antibiotic growth motor or a diet with an antibiotic growth motor. So if you look at the clean environment, the bird performance was essentially the same, whether the birds were fed antibiotics or not. So in a clean environment, there's really very little response to an antibiotic growth motor. If we look at the birds in the unsanitary environment with no antibiotic, there was a loss of performance. So a decrease in growth rate, a decrease in efficiency. When the birds in the dirty environment were given a growth-promoting antibiotic, performance came back up to the level of the clean environment. And so this is an indication that the growth-promoting antibiotics really are not working when there's nothing for them to work against. So in a very clean environment, low challenge, the antibiotics are really not doing anything. In the same experiment, they looked at interleukin-1, circulating levels of interleukin-1, which is a pro-inflammatory cytokine. And typically when the levels are higher, we take that to mean there's a greater inflammatory challenge. So in this environment, or in this experiment, again, when there was no challenge, so when the birds were raised in a clean environment, there was no response to the antibiotic. But in the dirty environment, when there was an antibiotic, the level of interleukin-1 returned essentially to the level of the control, but when no antibiotic was fed, there was more inflammation. So again, one of the ways that antibiotics work is by directly or indirectly reducing inflammation. Similar study looking at, in this case, it's a single commercial broiler flock. And the researchers looked at birds that were high body weight or low body weight. They took blood samples. And so the high body weight birds had low levels of serum lipopolysaccharide. And lipopolysaccharide is basically the component of gram-negative bacteria cell walls. And so the higher the level of lipopolysaccharide or LPS, the greater the translocation of bacteria across the gut and the greater the inflammation. So again, the high body weight birds had low levels of LPS, low levels of bacterial translocation, and the low body weight birds tended to have higher levels of LPS. And in that same experiment, the researchers looked at markers of inflammation. And again, the low body weight birds tended to have higher markers of inflammation. So growth promoting antibiotics really allow the birds to get closer to their genetic potential. There is no treatment that will allow birds to go beyond their genetic potential. So if we have a very, very clean environment with low challenges, the birds have a low need for inflammation, and they grow hopefully very close to their genetic potential. In a dirty environment, even if there's no clinical disease, what we tend to see is that there are often these little minor subclinical challenges, and we see a little bit of loss of performance. The birds get over it and they increase their growth rate. We might see some compensatory growth, but they don't get close to their genetic potential. And that continues throughout the life of the bird. And overall, it reduces their their efficiency. And through a number of studies, basically most of this loss of performance is associated with with systemic inflammation. And so if we can reduce that systemic inflammation, even the minor challenges that occur frequently, we can increase the performance. So this is some research looking at growth rate. These are two different experiments, and on the left-hand side of the screen, the challenge, the inflammatory challenge was heat-killed E. coli. So these are non-infectious cells, but the birds respond. The immune system recognizes it as a pathogenic bacteria. On the right side of the screen, the challenge was LPS, that lipopolysaccharide. So the control birds in each experiment grew fairly well. And then when the birds were challenged, there was about a 20 to 30% loss of growth rate. Now, in each experiment, the researchers also pair-fed birds, control birds, non-challenged birds, but to the level of feed intake of the challenged birds. So 20 to 30% decrease with the challenge. And when you fed broilers the same amount of feed as those birds, they actually grew better. Not as good as the controls, but they grew better than the challenged birds, and that was by about 9%. So looking at the total cost of inflammation, the total cost on productivity, most of that loss of performance is caused by a decrease in appetite, a reduction in feed intake, and we saw that on the previous slide. So about 70% of that loss of performance is associated with simply the birds eat less. But there's another 29–30% of lost performance that is not simply explained by a reduction in feed intake. So it's the systemic effects of inflammation that contribute to that loss of production, that other 30%. So what are the components of that lost performance? Well, there's a number of different factors, including there's an increased metabolic cost of the intestine. When there's an increased challenge, when there's an increased inflammatory challenge, there's an increased cost of maintaining and repairing tight junction function in between the intestinal cells. There's typically an increase in mucus production, and that mucus requires nutrients. It requires carbohydrate, it requires protein. And also the intestinal wall itself, it becomes thicker. So there's more tissue that has to be maintained. And of course, the intestine is a very metabolically active tissue. And so there's an increased cost to grow that tissue and to maintain that tissue. And that accounts for about 5 of that 29%. So the systemic cost of the immune defenses, the barrier functions. There are also changes physiologically in terms of tissue and protein synthesis. So in a healthy liver, the liver is producing a number of different proteins and sending those proteins out to the body. But when there's an acute inflammatory response, the proteins change. There's a large panel of acute phase proteins that are synthesized. Total liver protein synthesis increases dramatically. And actually, it requires a lot more fuel to not only build those proteins, but also to provide the energy to synthesize the proteins. And the liver increases in size as well. So there's a bigger organ making more protein, and so there's an increased cost related to that. Now, at the same time, so remember, feed intake goes down, and we have increased protein synthesis. So the amino acids have to come from somewhere. And so what we see is rather than a healthy muscle, breast muscle, for example, where in a growing bird, the rate of protein synthesis is much greater than the rate of degradation, in inflammation, we see that the rate of degradation is much greater than the rate of synthesis. And what's happening is those acute phase proteins are being synthesized with amino acids largely coming from the bird's own muscle tissues. So we see, if not a loss of muscle tissue, certainly a reduction in growth rate. Now, the other confounding factor is that when the muscle is catabolized, the amino acid balance of muscle is different than the amino acid profile of the acute phase proteins. And so if we look at cysteine as an example, with 1.88 times the amount of cysteine in a gram of acute phase protein relative to muscle, it means that the bird has to actually mobilize almost 2 grams of breast muscle or 2 grams of muscle tissue to provide the cysteine required to make 1 gram of acute phase protein. So it's an inefficient process, an inefficient way to provide the amino acids to the liver for acute phase protein. So with the reduction in feed intake and a reduction in amino acid supply, the mobilization of muscle tissue to provide amino acids, and the imbalance between the amino acid balance of acute phase proteins versus muscle, that inefficiency or those metabolic inefficiencies account for another 8% of that 29%. Now, we're probably all familiar with fever as a side effect of inflammation. If you've ever responded to a vaccination yourself, you know that you often have an increase in body temperature. And so there's some normal diurnal and within the day fluctuations in body temperature. But when the bird is undergoing a systemic inflammatory response, we see a much greater febrile response. And raising the body temperature is energetically costly. It takes additional energy above simply maintaining normal body temperature. And so that energy required for fever is another 3.5% or so of that, that lost performance. And then finally, the intestine itself changes. So the rate and efficiency of nutrient absorption that is consumed, the feed that is consumed, tends to be much lower. So if we look at particularly towards the bottom of this this table, things like lipids, pigments, minerals, but also things like methionine and cysteine are absorbed less efficiently. So same diet as when the bird is healthy, but the nutrients are much less efficiently absorbed from the digestive tract and for use by the body. So again, there's a reduction in feed intake, a reduction in total nutrient supply, and then the nutrients that are there are absorbed less efficiently. So the overall effect is a much lower level of nutrient absorption from the gut. And that's borne out, again, in this particular experiment, about a 9% loss in body weight gain associated with a systemic inflammatory response. So those digestive inefficiencies account for another 9%. And if you add those up, it accounts for about 25.5% of that 29% lost performance. So the majority of lost performance, first of all, is accounted for by reduced feed intake. And the other losses in the performance are largely explained by digestive inefficiencies, increased energy expenditure, metabolic inefficiencies, and the cost of systemic immunity. Just to put this into perspective, when the bird is not challenged, so when the bird is not having an active systemic inflammatory response, the amount of lysine in the immune system, so the immune cells in specific immune tissues, is about 15% of the lysine that's in a single egg, or about 5% of the lysine that's in one pectoralis muscle. When there's a strong inflammatory response, that amount of lysine in the immune system increases to about 1.2 eggs worth of lysine, or about 39% of the lysine in one pectoralis muscle. So there's a huge increase in the amount of, in this case, lysine dedicated to the inflammatory response compared to when the bird is not undergoing inflammation. Now, I don't have time to talk about a full list and details on alternatives to antibiotics, but I do want to mention a few specific ones, and I want to point out that an effective antibiotic alternative program will involve a number of different types of products with different mechanisms. Nothing works as well as antibiotics. But if we're not able to use antibiotics, we need to come up with effective replacements. And so we need to look at different mechanisms because we need to have different strategies that overlap and complement each other in order to have an effective response. So there are compounds that decrease microbial growth in the gut. So for example, organic acids, various phytogenic compounds. There are competitive exclusion products. So prebiotics and probiotics that make it more difficult for pathogenic bacteria to establish and replicate in the gut. There are products that inhibit pathogen attachment to the gut. So things like mannose oligosaccharides and xylem oligosaccharides and butyrate that reduce the ability of the pathogens to establish themselves in the gut. There are a number of products that increase gut integrity. So helping to maintain that gut barrier function, this would be things like butyrate and betaine and glutamate and glutamine. And then there are immunomodulators. So feed components or nutrients that help to have a stronger response in some ways, but a more efficient, a more rapidly resolved response. And this can include things like yeast beta-glucans and butyrate, various phytogenics and various nutrients. So this is not a complete list. And if you have a particular favorite product that is not listed on here, I don't need to exclude it. My point is that there are a number of different mechanisms that we need to be aware of. And usually, in order to have an effective antibiotic alternative program, we need to use multiple categories of these products. So in summary, immune function is critical for healthy birds. That means that they need to have a functional immune system. But when we look at what is good for production, a strong inflammatory response, particularly if it's responding to non-pathogenic challenges, can actually reduce performance. And so we want to minimize the negative effects of inflammation while still allowing the birds to have a healthy immune system overall. Inflammation is metabolically expensive, and adaptive immunity has a low cost. So we want to see a transition from innate to adaptive immune response as efficiently as possible. And in general, if we can avoid inflammation, this is probably the most effective way we can manage our birds. So rather than trying to have a stronger response to deal with problems, we want to minimize the need for inflammation in the first place. So with that, thank you very much, and I appreciate your attention.